My name is Jennifer Edgar, and my project was optimizing minimum inhibitory concentration assays for multidrug resistant gram-negative bacterial pathogens. Antibiotic resistance has been and continues to be a significant issue in medicine. Pathogens continue to adapt and obtain multidrug resistance, making it very difficult to treat patients with these pathogens. One method of resistance is through R&D efflux transporters. These transporters can often move several very different antibiotics out of bacterial cells and the periplasm, allowing the bacteria to survive. My project is focused on one such R&D transporter found in Neisseria gonorrhoeae, known as mtrCDE. The end goal of this project is to identify and develop inhibitors of mtrCDE in order to restore antibiotic sensitivity to antibiotic-resistant Neisseria gonorrhoeae. Potential inhibitors have been identified through computational means. This presentation reports our efforts to optimize minimum inhibitory concentration, or MIC, assays for multidrug-resistant gram-negative bacterial pathogens. After previously correcting the sequence for the mtrCDE gene, rapid colony transformation was performed as you see here to transform the corrected plasmid into a strain of Escherichia coli. The MIC assays were performed as you see above. Of course, a few factors were methodically tested to determine optimal conditions for the assay. These factors include IPTG induction time, the specific antibiotics used, antibiotic concentrations, and the amount of culture inoculated into the well plate. You see here the results of our efforts to select an optimal positive control. This control was obtained through the rapid colony transformation procedure presented earlier. At the higher concentration of Triton X-100, a detergent, not antibiotic, that is a substrate of mtrCDE, you can see that the positive control growth curve noticeably increases by the end of 20 hours. The negative control growth curve, however, does not increase. At the lower concentration of Triton X-100, both growth curves increase. This demonstrates that both the negative control and positive control bacteria are alive at the beginning of the final incubation period, as no growth would be observed without live bacteria. It also demonstrates that the MIC for the positive control is greater than the MIC for the negative control, and the difference between the two is very clear, making this positive control optimal for future MIC assays. Here you see the results of the optimization of final cell concentration for the assays. Clinical labs often report the use of the McFarland standard. We made the McFarland standard and measured its optical density, or OD, to be 0.11. However, it was uncertain whether this OD was to be for the mix to be plated or for the final mix in the well plate. So we tested both possibilities and found that 0.11 final OD in the well plate is optimal for these assays. For other labs, it is suggested to match the final OD to that of a McFarland standard. Here you see that at both high and low concentrations of ciprofloxacin, the growth curves of both positive and negative controls display similar growth patterns. There is no significant observable difference in the mix between the positive and negative controls, suggesting that ciprofloxacin is not a substrate of mtrCDE. So far, this research has produced an optimal positive control, an optimal final cell concentration, and the finding that ciprofloxacin is not a substrate of mtrCDE. These findings have improved the MIC assay. To continue this research, the assay should continue to be optimized by identifying the optimal IPTG induction time and negative control. In addition, experimentally obtained MIC values should be compared to literature values. Ultimately, the computationally designed potential inhibitors of mtrCDE should be tested to experimentally identify actual inhibitors. I would like to thank the SMU Center for Drug Discovery, Design, and Delivery, the Hamilton Scholars Program, Engaged Learning, and SMU Undergraduate Research for their support of this research.